Hello there! Today you are going to learn about meiosis, which is the other type of cell division. So write down these learning goals under Part A. Number 1, you are going to be able to explain the purpose of meiosis. Number 2, you are going to be able to describe how meiosis is different than mitosis. You might also want to write down these vocab words, which will be new, but you will know them by the end of today's notes. Meiosis, tetrad, gamete, haploid, diploid, and homologous. That last one is a real doozy. Homologous. Homologous. On this slide, you can see that there are several pictures of a cell going through meiosis. Do you notice what the main differences between meiosis and mitosis are? That's right, there are several more stages of meiosis because in fact the cell divides not once but twice, resulting in 4 cells at the end. And this is how your egg and sperm cells are made. So mitosis and meiosis are both types of cell division, but there are several key differences, and you will be asked to compare and contrast mitosis and meiosis on the next test. So FYI, you might want to make a note of that. So you've learned already that mitosis makes an identical copy of the cell and its DNA. It's like a clone of itself. It's asexual reproduction. Meiosis is different, however. Instead of making 2 cells, it makes 4 cells, and instead of them being identical, each cell is actually unique with different DNA. And these are called gametes or sex cells, and this is what makes up sperm cells in males and egg cells in females. Let's begin to look at the phases of meiosis. The reason that we reviewed mitosis is that the phases of meiosis are actually very similar. And if you're familiar with the phases of mitosis, that means you actually already know how meiosis works. We begin with PMAT again, but instead of it just being PMAT, we're going to put the Roman numeral 1 after each phase because meiosis is going to do it twice. So the first 4 phases are prophase 1, metaphase 1, anaphase 1, and telophase 1. There are many similarities as well because in prophase 1, the chromatin condenses to chromosomes. In metaphase 1, the chromosomes line up in the middle. In anaphase 1, they are pulled apart, and in telophase 1, 2 nuclei reform. Notice any similarities? One of the biggest differences in meiosis is that the cell divides twice. So after prophase 1, metaphase 1, anaphase 1, and telophase 1, the whole process is going to repeat again. And you have, surprise, surprise, prophase 2, metaphase 2, anaphase 2, and telophase 2. Each of the new cells made in the first phase of cell division is going to divide again. So again, the chromatin condenses into chromosomes. In metaphase 2, they line up in the center. Anaphase 2, they're pulled apart. And in telophase 2, the nuclei reform and you end up with 4 new cells. Let's now highlight some of the most important differences that makes meiosis unique from mitosis. The first and one of the most important events in meiosis is that in prophase 1, this crazy event happens called crossing over. And you can actually, under the microscope, see the chromosomes crossing over, and they form these special shapes called tetrads. So go ahead and write that down under Part D, tetrads. The prefix tetra means 4, and that's because there are 4 chromatids that are actually exchanging pieces of DNA with each other. That's right, they are cutting and pasting pieces of DNA with each other. One of those chromosomes is from mom, one of those chromosomes is from dad. And so in this phase, we actually are shuffling around the DNA. So here on the picture to the left, you can actually see a tetrad where the green chromosome and the purple chromosome almost look like they're doing a dance with each other, and their legs are crossed at the bottom because where the purple and the green are crisscrossed with each other, they are actually trading DNA. So go ahead and draw a picture of a tetrad at the bottom of page 1. And pretend the green chromosome is from dad and the purple chromosome is from mom. Look at the picture on the right. So after crossing over, they have shuffled pieces of their DNA, and when they're pulled apart in anaphase, shown on the right, you can see that mom's purple chromosome has a piece of dad's green chromosome at the bottom, and the reverse is true for dad. There's actually a piece of mom's purple chromosome there at the bottom. So they have shuffled pieces of their DNA. Go ahead and flip your notes over, and in little Roman numeral 3, it says crossing over between homologous chromosomes occurs during this phase, prophase 1. What does this mean? So go ahead and write this down. While in tetrads, the chromosomes will trade DNA. Now you know what crossing over means, and we're going to talk about its importance and what homologous chromosomes are in just a second. So why is crossing over important? After all, it seems quite risky to just mix and match DNA at random and shuffle the genes. It seems like a lot of mistakes could happen, and in fact, sometimes they do. So why would a species want to do this? Well, when we mix and match the genes between mom and dad, we are increasing the number of possible gene combinations that can occur in the offspring, meaning that every child that mom and dad have will be unique. This is why you don't look like your brothers and sisters. So crossing over increases the number of possible gene combinations. This means we are increasing genetic diversity as a species, meaning that it will be more likely that as a species we can survive in the event of a catastrophe such as a massive flu virus. So what does homologous mean? Homologous means similar. Homo is the prefix that means same, and you have two sets of chromosomes in most of the cells in your body. One set is from mom and one set is from dad. We have 23 unique pairs of chromosomes, but altogether in most cells we have 46, one from mom and one from dad. So homologous means that you have a chromosome with all the same genes from mom and a chromosome with all the same genes from dad. And even though they have the same genes, the specific DNA sequences that code for each gene might be slightly different. So in chromosome 1, let's say you have genes, like 4 or 5 genes on there. Usually you have a lot more. They have the same genes in mom and dad's chromosomes, but the specific DNA sequences might be slightly different. So they are similar. And so crossing over is going to occur between these homologous pairs of chromosomes so that you're shuffling genes between the same set of genes between mom and dad, but just changing the DNA sequences slightly. It's like shuffling a deck of cards. So it turns out that cells that have homologous pairs of chromosomes are called a certain term, and cells that only have one set of chromosomes are called another term. And we'll talk about that in just a second. Those are going to be called diploid or haploid. So we are now under part E, diploid versus haploid. Diploid cells actually are the ones that have two sets of chromosomes. The prefix di means two. These are 99% of all of the cells in your body, and this is what you've learned, that cells have 46 chromosomes. One set is from mom and one set is from dad. Haploid cells have half the number of chromosomes as diploid cells. And these are only your sex cells, the gametes, in other words. So they're going to be sperm cells in males or egg cells in females. They only have one set of chromosomes, so 23 chromosomes. So now you can go back and answer question number 6 above. Which cells have homologous pairs of chromosomes? Can you guess? That would be diploid cells, that's right. So how do diploid cells divide? Well, we've already learned this. Diploid cells are going to divide by mitosis to make an identical copy of itself and produce two daughter cells. Haploid cells, they actually really don't divide, but they are made by this process that we've been talking about through meiosis. Haploid cells are going to use meiosis to create egg and sperm cells. So one last important question. Why would our egg and sperm cells want to go through all of this trouble of dividing twice and reducing the number of chromosomes by half, from 46 to 23? Why would they want to do that? Well, think about it. These are our sperm and our egg cells. Eventually, during sexual reproduction, a sperm cell and an an egg cell are going to recombine to produce a two-celled embryo called a zygote, which will eventually develop into a baby. And if we didn't divide the number of chromosomes by half, every time an egg and a sperm combined, you would actually be doubling the number of chromosomes, which would be an entirely different species. And so we have to be able to reduce the number of chromosomes so that when they combine during sexual reproduction, you end up with a baby that has the same number of chromosomes as the parents. And therefore, the baby is still human. Take a look at these karyotypes that show pictures of chromosomes. On the left, we can see a picture of all the chromosomes from diploid cells, and on the right, we can see pictures of chromosomes from haploid cells. This is from a human because you can see that there are 23 chromosomes, and scientists often create these karyotypes to look for chromosomal abnormalities. Karyotype, by the way, which we've talked about before, is spelled Karyotype. So from these pictures, you can actually identify which karyotype came from diploid cells and which came from haploid cells. Remember, diploid cells have homologous chromosomes, so there is one set from mom and one set from dad. So you should see 2 copies for each chromosome, as you do on the left. In haploid cells, however, there is only 1 chromosome for each of the 23, and that's why on the right you only see 1 copy per chromosome. So this picture came from a haploid cell, which must have been either an egg cell or a sperm cell. One more really cool thing to note is that in karyotypes from diploid cells, you can actually identify whether it came from a male or a female because our sex chromosomes are determined by chromosome number 23. And in this karyotype on the left, you can see that chromosome 23 contains an X and a Y. So that must have come from a male because a female would have an X and an X at chromosome 23. So we're going to conclude with pictures of meiosis that are in order, top left to right. So in number 1, you can actually in prophase 1 see crossing over. There's little tetrads forming there. Number 2 is metaphase 1. They're lined up in the middle. 3 and 4 show anaphase 1. The chromosomes are getting pulled apart. And in number 5, you actually see 2 nuclei reforming in telophase 1. Number 6 would be prophase 2. The chromosomes are visible again, condensing from chromatin, and then they line up in the middle in number 7 in metaphase 2. 8 and 9 are anaphase 2, they're pulled apart, and in number 10 you can see in telophase 2, 4 new nuclei are formed, which are showing you 4 unique gametes. Well, there you have it, folks. That is meiosis for you, the other type of cell division that makes your egg and your sperm cells. Go ahead and write your 3-sentence summary. You will also need to finish Cornellizing these notes by adding 3 questions. And you might as well do it now so that you don't have to worry about doing it at the last minute before the test. Once you are done, you are ready to go ahead and start the meiosis modeling lab, where you are going to recreate this using gummy worms. Thanks for watching. Hope you learned something new.